Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Thomas Ichim. He's the CSO of Immorta Bio. So we're going to talk about senolytic immunotherapy, immunotherapy for cells that are old and called senolytic. So we're going to go into that. Dr. Ichim is a seasoned biotech entrepreneur. He's been in it over 20 years. He's launched and grown and realized the value of multiple biotech companies. He's renowned for his advances in regenerative medicine and immunotherapy. He's been a manager, a vice president, as well as president and CEO of emerging biotech startups and billion-dollar public enterprises. He holds a PhD in immunology and has published 130 more peer-reviewed articles than I have and authored 3 textbooks. So welcome, Thomas. Thanks for coming.
Thank you very much, Richard. It's an honor to be on your podcast. I've heard a lot about you, and I'm really glad to be here.
Oh, well, thank you. Well, tell me. So I hear about your background. You've been in biotech for a long time. I guess just just to start, what got you interested in biotech and in working in all these companies? What has become your goal, like your mission?
Well, I think my mission pretty much is to help people that are suffering. I mean, if you really think about it, life is pretty empty. Money doesn't really mean anything. The materialistic things, they come and they go. But at the end of the day, what you really leave is your ideas and your ability to make a difference in the state of human suffering and the state of human existence. And specifically how I got into this whole thing was when I was 9 years old, they did an IQ test on me and I scored very highly. And several years later, 2 years later, my mom was diagnosed with leukemia. So I was, it was terrible. But in a way, it was the best thing that happened to me because I focused all my life on cancer research, on leukemia. And as you know, Richard, the leukemia is a stem cell disease. So by studying leukemia, you study stem cells. So I was very active. My first peer-reviewed abstract I published when I was like 17. I got my first patent issued when I was 19. And, I've been in the field very aggressively from the point of view originally about saving my mom. And my mom passed away in 2010 when I was, I guess, like 30 something. And then after my mom passed away, I just became really obsessed with stem cells and with longevity and how we can apply concepts that we learn in longevity to actual diseases instead of just focusing on making people live longer. A clinical trial for a longevity product is going to take 20, 30 years and the investors don't like that kind of time frame. So one of the things I became obsessed with after my work in the area of oncology was stem cells, regeneration, and longevity, and how we can apply stem cells to very acute situations. For example, like COVID-19, we did some work and we published a paper on using mesenchymal stem cells to shut down with acute inflammation. We have some work that we're doing now that's very exciting that we'll get into. But again, to answer your question directly, I got into biomedical sciences because my mom got leukemia, and I wanted to help my mom and other mothers like that who were dying of this terrible condition.
Yeah. Were you able to help her, or was it just too early in your life, in your career?
No, it was, this is something, and my mom actually wrote a book about this. Unfortunately, it's in Romanian, so it's difficult to read. But no, what happened was back then they didn't have the internet, so me and my mom would go to the library. God bless my mom. I mean, usually I don't talk about this on interviews, but me and my mom and I would go to the Kitchener Public Library. This was up in Canada, Kitchener, Ontario, and we found out that there was a new treatment called interferon, and there's a new treatment for leukemia called interferon that they were administering in Toronto. Toronto was about 100 miles away. So we went to our doctor and we said, look, is there a way we could get on this clinical trial? And the doctor said, oh, it's far away. You shouldn't trouble yourself. There's not too much hope. It's highly experimental. But my mom insisted. And because she got on that treatment, she ended up living she ended up being stable disease on that treatment for about 10 years. So she got leukemia when I was 11 till about I was 21. She was stable. Then she stopped responding to interferon. We started doing something called placental vaccinations. And this may be of some interest to you. So placental vaccinations under the idea that the placenta is similar to cancer. And if you immunize against placenta, you can get antibody responses that cross-react from the placenta to the cancer. That caused my mom to go in remission for about 2-3 years. And after that, the kinase inhibitors came. Kinase inhibitors, a new class of drug. First kinase inhibitor, by complete coincidence, was designed exactly for my mom's type of leukemia. I didn't have anything to do with it. It was just God, I guess. But it was funny because, out of all the type of hematological malignancies, out of all the types of cancers, the first kinase inhibitor, which is a small molecule that specifically blocks the mutated protein, the first one was in my mom's leukemia, which was CML. And that gave my mom another 15 years. So at the end of the day, my mom ended up passing away when I was about 35, and she got leukemia when I was 11. And, when I was 11, they said she was going to die in 3 to 4 years. So it just shows you how important science is and medical research, and how we have to be advocates for the importance of funding medical research and funding the biotech industry. Because if it wasn't for medical research, she would have just died in 3-4 years. When she got leukemia, my youngest sibling Matthew was 6 months old, and she was breastfeeding him. Now Matthew is an accountant at KPMG, and she got to see Matthew grow up. She got to see Matthew get his CPA. And it was just something. So that's the power of science, and that's something that motivates us. And being in the biotech industry, sometimes we forget why we're here. But every time I hear stories like this, or every time I think about the reason why we did get into this field to begin with, it's something that sets apart this field from I think any other type of human activity.
Yeah, no, that's great. Hey, your mom had about 20 more years than she otherwise would. That's fantastic.
Exactly. Yeah.
So what is your current work about? Let's fast forward to that and, talk about some of the nuances of it.
So the current work is very interesting. It's a very interesting concept. It's basically how to fight aging, fighting diseases of aging, and fighting aging as disease. So what exactly that means is we have 2 platforms to fight aging and to fight diseases of aging. First platform is to kill all cells. This is called senolysis, killing senescent cells. So this is very exciting. And this all came to news to me. So about 8 years ago, a guy at the Cleveland Mayo Clinic called James Kirkland, he found out that if you kill senescent cells, you start to rejuvenate the body. So very peculiar. So senescent cells, just to give it a little bit of a background, when a cell multiplies at about the 50th cell division, it undergoes what's called senescence, which is it becomes a zombie. It doesn't die, but it doesn't really live either. What I mean is, it sits there, and originally we just saw these cells accumulate and do nothing. This Dr. Kirkland and people after him found that the senescent cell starts to produce different senescent-associated secretory phenotype proteins. What that means is, once the cell reaches this critical 50 doubling, it produces all these poisons that poison the cells around it. So aging is an infectious process, infectious senescence. How do we know this? Because he published papers where he would take cells from mice that are of human equivalent of 80 years old, and put those cells into the fat pad of mice that are human equivalent of 20. Those mice would start accelerated aging.
So is that because they're very active in cell-to-cell signaling, and so maybe they drown out or obfuscate normal cell-to-cell signaling and cause things to act in their own way?
That's a really good idea. And again, this field is moving so quickly that it might be what you just said. What we do know so far is this. They secrete inflammatory cytokines. Specifically, they secrete a lot of interleukin-6, interleukin-11, interleukin-23 receptor. These inflammatory cytokines induce accelerated aging by stimulating inflammation. Inflammation causes oxidative stress, causes activation of neutrophils, monocytes. Those things produce free radicals, which then tell the cells around the senescent cells, the young cells, they accelerate their aging. So it's for production of inflammation. And we know this because if you take senescent cells and you block these senescent-associated secretory phenotype proteins, then they do not accelerate senescence. But here's the crazy thing. In the major publication, I'm sure there's a lot, but in the major one that a lot of people cite, what they did was they measured grip strength of the mouse, and ability to dangle from a structure. The older mice, they lose grip strength, just like humans lose muscle mass. When you would kill the senescent cells with what they call senolytic compounds, you would regain the grip strength. How do people kill senescent cells? They kill them with something called, the original one is a combination. You're going to love this. My life is built with all sorts of weird coincidences. The drug they used to kill senescent cells was a drug that my mom used. It's a leukemic drug called dasatinib. And they found this, Dr. Kirkland found that dasatinib, we still have some dasatinib at home, by the way. But what they do is dasatinib combined with a natural drug called quercetin. This combination of dasatinib and quercetin induces selective killing of most of the senescent cells. And how it does this, by the way, is because the senescent cells rely on a protein called BCL2. See, BCL2 stops cells from dying. So when a cell decides it's going to be senescent, it upregulates this BCL2 so it doesn't die, but it doesn't live. And what I mean by doesn't live is it stops performing its usual function. So for example, a liver cell's function is to have cytochrome P450 and detoxify stuff. A senescent liver cell still hangs around, stops detoxifying stuff. It just sits there and gathers space. But then what Dr. Kirkland found out when people after him is that it spits out this toxic stuff. So here's the bottom line. If you kill senescent cells, and there's so many publications, they're coming out at a geometrical rate. If you kill senescent cells, you improve a lot of the human chronic conditions. Big peer-reviewed papers published, for example, disc degeneration, like lower back pain, where the nucleus pulposus cells that maintain the cushion in your disc, they start atrophying. A lot of that is senescence. If you give senolytic inhibitors, you prevent the onset and progression of disc degeneration. If you give senolytics to a mouse that you give a lot of alcohol, you prevent the cirrhosis. If you give senolytics to a mouse after stroke, you reduce the infarct volume and you accelerate the neurological, the brain function coming back after a stroke is accelerate recovery.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
How do you administer this? What if you did an infusion of quercetin and dasatinib?
Yeah, you don't even need to
through the blood.
You just take it orally.
Well, why not do something like, like if you don't know, I would think, not just one cell type becomes, senescent, and at different rates, I'm sure they do in different people. So if you administer through blood, would it not preferentially reduce the senolytic cells in various body systems and organs as they transit?
It does. Absolutely. You're absolutely right. That's exactly what it does. But here's the catch. The catch is it doesn't work that well. I mean, it works well, but first of all, dasatinib, you got to remember still it's a cancer drug. It's got its own toxicities. BCL2, the gene that they're targeting, BCL2 is an anti-apoptotic gene, which means it stops cells from dying, but it's also found in healthy cells too. So the dasatinib quercetin combination, which is good and has helped, it helps, but it's not a complete magic bullet. So this is where we come in. And by the way, so they are doing clinical trials with dasatinib and quercetin. They had some beautiful data in idiopathic pulmonary fibrosis in humans. It's a terrible disease where you get scarring of the lungs. As the name implies, no one knows why it's idiopathic, but people found out it is the pathology is associated with accelerated senescence. And the dasatinib quercetin had some good effects. There's also another drug, another senolytic that everybody uses called ABT, 263. It's an anti-lymphoma drug, and it's got its one set of issues. But so here's what we do. We always say it's better to let the body heal itself, or to leverage what the body does. So if it's okay with it, we'll take a quick step back. If you think about stem cell therapy, what is the only stem cell therapy that really works? There's only one stem cell therapy, which is hematopoietic transplantation, bone marrow transplant. Bone marrow transplantation, what you do is you are not asking the bone marrow to become a cardiomyocyte, or you're not asking the bone marrow to replace your liver. All you're asking the bone marrow to do is do what it normally does, which is make blood. Except you're telling it to make blood not in the same person, but in a different person. So for example, if I had leukemia, what they would do, because you and I are brothers, Richard, what they would do is they would give me a lethal dose of radiation and chemotherapy to kill all of my blood-making cells, because what that does is it kills my leukemic cells, but also kills the blood-making stem cells. And then what they do is they rescue me by giving me your bone marrow. And your bone marrow knows to make blood, so it gets a little confused. Sometimes it might attack me. It's a little thing called graft versus host. But for the most part, the bone marrow transplantation has cured tens of thousands of people with leukemia. Now, the first company that I made and sold, I used that same principle, and I said, look, the blood vessels get made in the woman's endometrium every month. Why don't I just take that same cell that makes blood vessels in the woman endometrium and put in the muscle of ischemic diabetics to increase blood supply? And we did that. We made a company called MediStem, got FDA clearance to begin clinical trials, and then we sold the company to Intrexon. So now we're doing the same thing. Now we're saying to ourselves, what is the body's normal way to get rid of senescent cells? What it is, it's the immune system. The immune system has different mechanisms that it uses to recognize and kill cells once they become senescent. The problem is this. If that's the case, why do we get old? The problem is that, well, two reasons. One, the immune system gets old. Once the immune system starts to get old, then it cannot kill the senescent cells itself. So these are little books.
That's why, to do this as close to naturally as possible, you want to remove the senescent cells from the immune system to de-age it, and then it will take care of everything else.
Exactly. Well, yes, that's the idea. How we do this is basically we revive the immune system by using an approach that we've used before and other people have used before, which is called dendritic cells. So with dendritic cells, these are the most potent immune activating cells. So dendritic cells, they usually live in the skin, but you can find them anywhere. But what they do is they sit around and they gobble up different pieces of protein. If they gobble up a protein that is dangerous to the body, then they get activated, and then they in turn instruct T cells to kill it. So the dendritic cell, it has been used and has been FDA approved for treatment of prostate cancer. So people with prostate cancer, what they do is they take their blood, they make dendritic cells, and they make dendritic cells that have a special protein found only on prostate cancer cells, a PSMA, prostate-specific membrane antigen. So they take this PSMA, I believe it's PSMA, or it's PAP, PSMA, I think, and they put it on the dendritic cell, and then the dendritic cell activates the T cell to go and kill prostate cancer. We are doing the same thing, except instead of using prostate cancer, we're killing senescent cells. And instead of using that protein, what we do is we make senescent cells from the person, from the patient. That way we have a personalized anti-senolytic immunotherapy. How do we do this? We take a little skin biopsy, and from the skin biopsy, we grow specific cells. Those cells are the same cells that surround the tumor. See, when tumors start growing, they produce this scar tissue around them that's primarily senescent cells. So we mimic that process in the lab by taking a skin biopsy and growing these cells. Then we take the blood from the patient and we grow dendritic cells. Then we take it a 2-to-1 ratio. We take the senescent cells, we kill them, we feed them to the dendritic cells, then we put this in the body. Once you put this in the body, then the immune system goes crazy. And so how far are we? So we have 20 patents filed and pending, and we have demonstrated in animal models. We have one peer-reviewed publication, showing that if you administer this senescent cells that have been fed to dendritic cells, if you administer this combination, we call this Senovax. If you administer this to mice with lung cancer, the lung cancer goes into remission. So we've demonstrated that. We have announced by press release, but we have not published yet. We're in the process of publishing some mechanistic data on specifically how this works, how the dendritic cells that we fed senescent cells program the T cells to go kill the cancer. Very exciting thing is that we've observed that this works not only in lung cancer, but it also works in pancreatic and glioma and breast cancer. So we're very excited about the data. We're in the process of going through the FDA And the exciting thing is there's something exciting that we've announced, we haven't published, we will be publishing this soon. If you, as you pointed out with quercetin and dasatinib, if you're going to give a senolytic, something that kills senescent cells, if you're going to be giving that to the body, you're going to see all sorts of senescent cells getting removed, not just the ones surrounding the cancer. So this is something that has us very excited.
When you say administer, do you administer by injection or pill or?
We administer by injection, by intradermal injection. And so what it does, you do not need this to go through the whole body. You just need it it's just like a vaccine. When you inject the vaccine, you put it here, but the effects are systemic because the antigen-presenting cell, the dendritic cell, goes into the lymph node, and that's what stimulates the T cells, and the T cells go and circulate everywhere, and the B cells go and make antibodies that circulate everywhere. But the exciting thing is this, that this anti-senescent immune system not only seems to inhibit the cancer, but it stimulates multiplication of bone marrow cells. So what we believe is happening, so after, if you take bone marrow, but the experiments what we did was we would take mice, we'd immunize them, and then we would give them chemotherapy to kill off the bone marrow. When the bone marrow comes back after being killed off, when it recovers a lot quicker in the mice that received Senovax. And we believe that this is because there are senescent cells that are preventing bone marrow from coming back after radiation. And it was not just that we believe this, but people have published that if you give small molecule, if you give quercetin and dasatinib after radiation, the bone marrow accelerates quicker. So we're seeing this Senovax, we're seeing it not only as potentially as a cancer therapy, but also to be able to reduce some of the senescence that has come into the body as a result of other cancer therapy. If you look at somebody who's gone through chemotherapy, you can tell they look 10 years older. And that's because this published chemotherapy not only induces dying of healthy multiplying cells, but cells that are not actively multiplying. Chemotherapy can induce senescence.
Question here. When you're harvesting the blood from somebody, do you induce any kind of fever or sickness in them to kind of ramp up the immune system? And then when you're harvesting the blood, it may have a lot more immunological components in it. And when you make it, when you refashion it to be re-injected, maybe it'll be more efficacious. I don't know.
A very, very good point. And, they do this, for example, they do this in certain protocols do this. What we do is once we have the blood outside of the body, that's where we activate it. So we take the blood from purified, we take out the white cells. So we spin it down. I don't know if Richard, if you've ever seen this, but you put the blood and they put a little density gradient on the bottom of the tube. And then what happens is you have a layer of only white blood cells. We collect a layer only of white blood cells, and we put in a special media containing several cytokines. That media activates the dendritic cell stem cells, the dendritic cell precursors to multiply. So we get a lot of these dendritic cells. Then we feed the dendritic cells. We feed them the senescent antigens. Then we make them go crazy by stimulating them in vitro. That way, we don't need to get the whole person sick. But to your point, the protocols you're talking about, you may remember at the turn of the last century, there was a physician called William Corley. And what he did is he would purposely get people sick to ramp up the immune response. And that causes almost the same amount of remissions as checkpoint inhibitors do today. So there definitely is something for hyperthermia and for inducing the whole body sickness. The problem with William Corley's approach was that he could not control it and some people died. But, you see this whole sickness too, like even with, the modern day name for it now is cytokine syndrome. And, now we're a lot better at controlling it than we were back then. But yes,
okay.
The sickness is definitely something. It's one of those strange cases where the sickness could actually be a good thing.
Have you used this in animal models? And what about organoids too?
That's a very good question. No, that short answer is no. No, we have not. It's difficult. It's difficult with organoids because you would have to have an immune system in the organoid and to have that interaction going. But
yeah, that's true. You need to at least have chained organoids, like heart chained to liver, chained to other organs to see.
Do they have that?
And some people I've spoken to, yes, they're starting to do that because you may not see toxicity, let's say in the heart, but then the liver gets it. Now it changes it to a compound that then becomes toxic downstream to the kidneys, let's say. So that's why they're doing, from what I've seen, chained organoid systems now.
I've never heard it. See, that's a beautiful thing with that's why I always wanted to have a podcast, like I was saying before, because the amount of cool stuff that you learn, Richard, must be just amazing. Yeah, I've never heard of this word chained organoid. I will look at it. But yes, I've got an approval. So going back to the story about my mom, how she was taking the placental vaccine, what happened was after after I sold my company Medistem, after I sold the company that made stem cells from the endometrium for the diabetic feet, I had some money on my hands and I said, what do I want to do with my life? And I made a company where we actually got FDA approval to do clinical trials on placental vaccines, very similar to the one my mom got. The one my mom got was considered alternative medicine. It was available only in Bahamas, and it was completely uncharacterized. So what we did is we did a deep dive into the cellular mechanisms of action. The reason why I'm saying this is we have gotten FDA approval. Me and my partner, Boris Resnick, we had a CRO where we would do clinical trials for other companies, but we also had our own clinical trials. So we've gotten many FDA approvals, and we're very confident that all we're going to need right now to go into humans, we're just going to need a little bit more animal safety data, and the FDA will allow you. The FDA is moving more and more towards organoids, but these kind of things are more for small molecules. See, with small molecules, like you said, there's all sorts of different toxicity issues. But when you're doing immune modulation, one of the things with immune modulation is, to date, from what we have seen, and we have had regulatory consultants look, there really are no organoid type models that represent the functioning immune system that you can connect to the multiple different organs and have something in vivo. There's humanized mice, and we do have some data on the humanized mice where you put human tumors and human immune cells and the human vaccine. But yeah, right now we are not using organoids.
Okay. So how long does this immune boosting effect last? Like, what is the profile of it? Does it cause, again, a cytokine storm that needs to be controlled? Like, is there a big bump up in activity and then it slowly goes down? Or, like, what does this look like in people? And do you need to control the administration so it works better instead of maybe just one big pulse?
What happens is that this is antigen specific. So for checkpoint inhibitors, where you see this cytokine storm, what you're doing is you're taking the brakes off the whole immune system. So checkpoint inhibitors, there's molecules like CTLA4, PD-L1, and these are inhibitory molecules that block any kind of immune response. Once you block the blockers, when you give antibodies to these, you have the whole immune system going crazy. With what we're doing, we are only stimulating immunity against senescent cells. The senescent cells that we lysed, that we took from the person's skin, we expand the cells, we make them senescent. So because the immune response, because the senescent burden of the body is a lot smaller, we have not seen any kind of cytokine storm at all. We have done extensive studies on cytokines. What we see when we inject mice with the Senovax is we do see a reduction in some of these senescent-associated proteins. So, for example, which indicates that they're reducing the number of senescent cells. But in terms of cytotoxic reactions and cytokine storms, we have done multiple immunizations. One of the things, because we've gone through the FDA, one of the things the FDA always asks you to do, and we've done these studies, is to inject at twice, 2 times, 4 times, and even 10 times the dose that you plan to be going into humans on a per kilogram basis. And we've done this, and we have seen no excessive immune activation. Again, the reason is, if you look at dendritic cell therapies as a whole, so the dendritic cell therapy, you feed the dendritic cell whatever you want to get the T cells to get mad and kill. So, for example, if you want to immunize against herpes virus, you take herpes virus proteins, you feed them to the dendritic cell, and you inject those dendritic cells. Because you're inducing immunity only to one type of antigen, you never get systemic cytokine syndrome or the cytokine storm. You get the cytokine storm when you use checkpoint inhibitors, and sometimes you get it in CAR-T. With CAR-T, it's a little different. With CAR-T, you're taking all of the cells, or a lot of the cells, transfecting a very strange construct that the body's never seen before. The human body in all of history has never seen this T cell with a B cell receptor on it. That's why they call it CAR, chimeric antigen receptor. So, the body's never seen something like this. And when you introduce something that the body's never seen as completely genetically modified, and you're modifying the whole population, you see the whole population of T cells, you see some strange things happening, especially in CAR-T, because the CAR-Ts initially, the ones that work really good are against CD19 on B cells. Basically what happens is this kills all the B cells. So you have a person walking around without the second, we classical immunologists, we think of the immune system as being T cells and B cells. People with lymphoma, after you give them CAR-T, they don't have B cells anymore for the rest of their lives. And, people at the beginning were horrified, but the hematologists were saying, hey, if you have this type of B cell lymphoma, you're going to die in several months. Better to live without B cells but be alive. It's just like the plus or minuses of organ transplantation. If you need a donor kidney, if you don't take immune suppression, that donor kidney is going to die in several weeks. But you're going to be alive if you take immune suppression. You just have to be careful make sure you don't get infections, and with antibiotics and supportive care. So it's always a dance, but
I have a way out thought that just came to my mind. I know it's not really in your purview, but is there any senescence in microbiome that people have? Has that been observed, and how does that interact with our somatic cells?
see, this is why I enjoy your line of questioning, because microbiome can induce acceleration of senescence. And from the other point of view, senescence affecting the microbiome, I don't know if anybody's even looked at that. I'm gonna check. We might have to name you as a co-inventor on one of the
Yeah.
patents. This is a very strange, but very logical idea. So with what we do about the microbiome, I'm sure you're very familiar with these studies. If you take the microbiome from somebody who's lean and in great shape, and you transfer it to somebody who obese or metabolic syndrome, you see an active protection and a reduction. And then when you look at the senescent cells, so if you look at the senescent cell burden of somebody who's obese, after they've taken microbiome from somebody who's lean, you see a decrease in the senescent cell burden. So the microbiome, we know, can definitely affect systemic levels of inflammation. And those levels of inflammation, they can definitely change the microbiome. But the microbiome stimulates inflammation, and the inflammation can stimulate senescence. That's well known. But can the senescent cells promote outgrowth of specific pathological microbiomes? And that's a question that, to my knowledge, nobody's addressed. But I'm going to look, because that's a very, very interesting question. Because let me give you an example. I saw you have a Twitter, and I followed you on Twitter. My Twitter is @exosome. And I was just tweeting about several days ago. I was just tweeting that, oh shit, it didn't let me follow but I'm going to try following it again, because I've reached the maximum number of people I can follow. But I'm going to try following it again, your podcast. But the point is that I was tweeting about microbiome. If you have dysbiosis and you administer stem cells, the stem cells don't work. There's specific butyric acid and certain metabolites made by the microbiome that when you're dysbiotic, the stem cells will not work. So that correlation is a very interesting correlation. And as you know, the more stem cells you have, the less senescence you have. So microbiome stimulating senescence, definitely there's rationale for that. But the senescence cells messing up the microbiome, that's something where we have to look like.
Here's why. I'll just give you a couple quick threads that are coming together. So a long time ago, I interviewed Florencia McAllister. I forget where she is, but they observed differing microbiomes locally around the tumors of, in a pancreas. Normal pancreatic tissue had a certain microbiome. The tumor-laden pancreatic tissue was different. And you talked about the cell membrane, different proteins are expressed senolytic cells, so therefore they would attract a different microbiome to them. And then you also said like tumor cells will have a senolytic or senescent cells around them as a border, as an outside border. Yeah. So that's probably why it changes their interaction with the microbiome and a different one forms, because there's different molecules now being exchanged. So I would think there is a big interplay in all this.
No, I think definitely, and I think the tumor microbiome question is just fascinating. And for example, there was, I believe his name was William Reif back in San Diego, and William Reif was talking about these microbial type particles that I believe he called them somatids. And you could see these in the blood, but you could only see them with a special microscope. And, everybody said he was crazy. But now we understand, with the microbiome revolution is all because we can only grow and culture 0.001 of everything in the microbiome. So once we had the ability to see this whole zoo, the way I think about it as a zoo, because there's all these different species, we're only able to see them through with 16S ribosomal sequencing. You cannot culture those things. To the microscope, a lot of them are too small or you just can't see them. So now that we understand the microbiome, and this is a whole revolution, because as you may have seen in the studies, I believe it was New England Journal of Medicine or Lancet, there was a study where they looked at checkpoint inhibitors. People who are non-responsive, people who were on antibiotics and had gut dysbiosis, they lived something like 4 months on checkpoint inhibitors. People that did not have antibiotics and were not dysbiotic lived like 17 months. These are huge differences. And the microbiome, this microbiome revolution, I still believe to some extent is we're only scratching the surface. And from what we know, black and white, it's not being implemented. You go to the oncologist, you ask about dysbiosis, they laugh at you. They say it's nothing to do with anything. Well, that's wrong. It has a lot to do. It even has to do with chemotherapy resistance, the microbiome. So, think about this. 100 microbial cells, at least 100 to 1,000 microbial cells in your gut for every mammalian cell that you have. That's why they call it the second genome, right? Very interesting stuff.
Okay. So what do you expect that you're going to be able to make a breakthrough on in maybe the next couple of years?
The breakthrough is this. So there's two things. So we talked about this analytics. Analytics, we hope to get the FDA clearance this year and have a clinical trial completed probably 2026. And that's going to allow us to IPO 2026, 2027. You know, we IPO'd two other companies, Creative Medical Health Technology. I took that on NASDAQ, and Fibrobiologics. I was the chief scientific officer. We went to NASDAQ. So all of these technologies, cell-based technologies, this one that we're doing now is very exciting. Breakthrough number one is going to be a completely new first-in-class way of treating cancer as a monotherapy and as an adjuvant therapy to current immunotherapies. You know, one of the most exciting things we're looking at is CAR T cells still have not penetrated solid tumors. And that's because, in our opinion, the senescent cells, our Xenovacs were excited about the possibility of potentially unlocking the potential of CAR T into solid tumors. The second thing that we did not talk about is the cellular rejuvenation program. And the cellular rejuvenation program might be even more interesting.
Yeah, I figured there's going to be a big group of people that hear this and they say, ooh, I want this treatment every 6 months to stay young and healthy and all that.
Yeah, we're not going to be able to do that because it's highly regulated through the FDA. You know, this.
Oh, no, I know, but I'm just saying, believe me, people are going to be interested. And
oh, yeah.
There'll be offshore outfits popping up that'll for this and all that probably.
Yeah, no, it's definitely possible. Now, the other thing is the cellular rejuvenation program. Cellular rejuvenation program, what we do is we take our blood, and we've done this from a 76-year-old person. We've done it from multiple people. We take out adult blood, and from that blood, we generate an immortal cell, a cell that never dies. That cell is similar to an iPSC cell. We call them personalized regenerative cells. So what this is we take from the blood what we call a mutable stem cell. We have a special population that we have IP on. We expand that cell, and from that we take it and we make it into like an iPSC cell, a very early pluripotent cell. From that very early pluripotent cell, then we bank it, and we can make an unlimited supply. The beautiful thing, so people have published this, but the biotech field more or less hasn't even gone on to this. A pluripotent cell can be multiplied forever and does not develop mutations. So with that, we're making several things. Our lead program is liver failure. We're making hepatocytes. Liver is a multi-billion-dollar disease, liver failure. Right now, the only two options are liver transplant or hepatocyte transplantation. Hepatocyte transplantation is basically the single hepatocytic cells. This has been demonstrated to cure liver failure. It's just we don't have enough hepatocytes. We've already manufactured scalable number of hepatocytes, show that they work in two models of liver failure, carbon tetrachloride and acetaminophen-induced liver failure, and now we're pushing this towards the clinic. The other thing that's even more exciting is mesenchymal cells. Mesenchymal cells are autologous repair cells. They're repair cells. They can repair a variety of tissues. Right now, the big problem is, so mesenchymal stem cells got approved by the FDA in January of this year for